Hello, and a very warm welcome to this Cell and Gene Therapy Insights webinar, Accelerate Process Development with At-Line Concentration Measurement. I'm Charlotte Barker, an editor at Bioinsights, and joining me today is Joe Ferriolo, Associate Director of Bioanalytics at Repligen. Joe leads the Bioanalytics Applications team and is in charge of the SoloVPE Variable Pathlength Spectroscopy System for at-line applications. Joe's been with the company for more than 20 years, with over 10 years of development and validation experience in analytical applications. Today, Joe will explain how the CTech SoloVPE system, when used at line in one or more stages of a process, can help quickly identify process characteristics, provide key insights, and allow for process optimization in minutes. First, we'll have a presentation from Joe, followed by a Q&A discussion. As always, do feel free to ask a question at any time throughout the event using the Ask a Question box, and we'll try to get to it during the session. And with that, I'll hand over to Joe for today's presentation.
Hi, and thank you for that introduction. My name is Joe Ferriolo. I'm the Associate Director of Bioanalytics for Repligen. Today we're going to talk about accelerating process development with our at-line system for concentration measurements, mainly the SoloVPE. Repligen has a product line that's been well in the industry for several years. We are the analytics portion of that product line with the SoloVPE for at-line measurements and the FlowVPX for in-line measurements. In the future, you'll see pairings of this technology together for integrated solutions, but today's discussion will be primarily around the SoloVPE and at-line analytics in the lab. The current areas within the downstream process that we're looking at are from UFDF through the affinity columns into chromatography. We are interested in seeing how we can use slope spectroscopy to reduce the number of cycles, reduce the amount of raw material required, and accelerate the time to market. In order to do this, we're going to fundamentally need to change how UV is typically done within these process steps. It is our hope that through our experience and leveraging our technology, we can reduce the error and the cost and provide a more robust solution for these process steps moving forward. For one example, the current AAV quantitative analysis methods that are typically used have acceptance tolerances for accuracy and repeatability that frankly make all of us feel very uncomfortable. Anything with a plus or minus tolerance of double-digit values, especially up to 20 to 40%, based on whether it's a qPCR, ddPCR, an ELISA measurement, or AUC and TEM, provide challenges in understanding what the actual values are, and being able to refine the method itself makes it problematic to understand where that error is coming from within the given methods chosen to run the assay. We've done some work to help understand where these challenges exist, how we can influence in changing them, and hopefully provide a comparable solution to the industry to allow for a very quick determination of tighter concentration in minutes, not days and weeks. Before we continue to case studies and examples, it's pertinent to give you an understanding of what slope spectroscopy is and why our form of UV is different from anything else in the marketplace. Traditional UV measurements require a fixed path length cell to be used for analysis. This means the concentration has to be the variable in our equation. Introducing dilution error in limited ranges of spectrophotometers make it very problematic to define this as a platform method for most industries using to leverage any form of UV technology for concentration determination. The amount of time, the amount of error associated with dilution doesn't necessarily make for a robust measurement. Our solution to address UV is making the path length the variable, so the concentration stays fixed while the path length is the variable. So you can see in the graph on the right, our path length is determined where the light emits from our fiber optic light source to the bottom of the sample vessel. The path length ranges from 5 microns up to 15 millimeters and as small as a 5 micron step. This gives the SoloVPE the option to scan up to 3,000 different choices of path lengths to find the best linear regression data it can find within the sample measurement itself. Therefore, because we don't need sample dilution, we're able to measure most high concentration samples neat without the need for any sample manipulation or dilution error. The second part of slope spectroscopy is we do not rely on a single absorbance value to calculate concentration. We rely on multiple absorbance values to plot a slope regression, which shows the change of absorbance over path length. In the top right graph, you'll see each of those data points sitting on the regression line represents an additional path length of data. It's also represented spectrally in the bottom right graph. Each one of those spectra represents another path length of data. This allows us to actually quantify the regression line with an R-squared value of 3 nines or higher. So we've now removed all of the sample manipulation from the process and proved linearity with an R-squared of 3 nines or higher with a 1-minute test asserting a pass-fail value based on the validity of the R-squared value. The way that the system works is there's an algorithm that monitors the change of absorbance from path length to path length in the initial search, looking for one absorbance values. Once the software determines what path length is closest to one absorbance, it will then stage 10 data points down to create the slope regression. Each acquisition takes less than a minute, and it's independent of user operating the system. So we are still very much working off the Beer-Lambert law equation. However, except for the absorbance and path length portion, we only care about the change of absorbance. So now it is slope divided by extinction coefficient, which will calculate your concentration. This has allowed us to significantly reduce process steps for simplicity and speed. A traditional UV process is usually defined by 7 steps, from estimation and dilution all the way through reporting, which takes anywhere from a half hour to 180 minutes, while the SoloVPE has been refined to a 2-step analysis, measure and report. So we're removing or automating all of these multiple steps to gain time, introduce robustness and simplicity to this OD measurement, GMP compliance, electronic data signatures, electronic data storage to simplify a process that can be used in GMP environments without any sample or user manipulation of sample volume or data at the end of the process. Some of the other areas that we're looking at outside of protein concentration determination will focus on the next few slides related to gene therapy applications, mainly evolved around plasmid purity, DNA, and viral titer. This is a simple list noting where some of the other SoloVPE and FlowVPX methods are being used to give you an idea of just where our technology can be implemented. If the former or current method is a UV-based solution, you now have the option to implement slope spectroscopy in either configuration for at-line or in-line with any one of these applications. Now we'll talk a little bit about viral vector production as well as some other gene therapy related applications based off some testing and studies we've done internally and with collaborative customers. Some of the main challenges we have with introducing our technology into the current market is understanding where the error associated with these particular methods are coming from. As I mentioned earlier, tolerances ranging from plus or minus 20% to 40% don't exactly rely on a robust method. And while some of these methods technically might be providing you an estimation of values, we are interested in understanding, 1, can we be comparable to these methods? And 2, can we leverage our technology to provide a more accurate solution to the market? So the data that you will see in the coming slides demonstrate how the SoloVPE system is equivalent and also give you some insight to just how well the technology performed versus customer provided samples and NIST traceable standards. This first case study was a collaboration with Pfizer for determination of plasmid DNA purity in human gene therapy products. What we've done is we've taken 25 different levels of insulin and compared the theoretical purity ratio versus what the SoloVPE measured. The SoloVPE technology will take a dual wavelength slope measurement for 260 for the DNA, 280 for the protein, do all of the calculations within the validated software, and provide us that purity ratio as the output. Not only were we comparable with differences less than plus or minus 2%, we also ran this a second time on purchased material through a third-party vendor and achieved the same results. The former method relied on a fixed path length technology that did not provide the resolution to measure high concentration standards. So it relied on dilution error in order to make the measurement, which introduces time and error to the process, where this multi-slope wavelength analysis can be done in less than a minute. The second case study is for antisense oligonucleotides. This collaboration was Ionis Pharmaceuticals, and they were interested in leveraging a UV solution for very highly concentrated oligonucleotides. Not only were they able to use our variable pathlength technology in their process, they were able to move away from an HPLC method and use something simple like a UV method, because our solution does not require any form of dilution. More importantly, this project was going to be developed and validated and ultimately transferred to 2 of their contract manufacturers. This data represented in the table shows the in-house percentages of recovery compared to CMO X and CMO Y. One of the CMOs is in the United States, the other CMO is in Europe. And between all 3 companies, the differences between the in-house samples versus the transferred method on SoloVPE were all well within plus or minus 2%. So not only do they gain the benefit of moving away from HPLC, which implemented a 67% cost reduction, but they simplified the process and have a robust way to transfer slope-based methods with collaborators and partners within the industry. The technology does not rely on the skill set of the scientist any longer in relation to sample prep and analysis. Therefore, the validity of the measurements based off the R-squared values of each slope regression provide all parties with the evidence that they have accurate and repeatable results from site to site and organization to organization. Saving the best and certainly most interesting for last, AAV viral titer concentration. The 4 current methods being used, being either qPCR and ELISA, ddPCR and ELISA, AUC, and TEM, are the 4 current industry standards for this particular measurement. Most companies have their preference for which technology they believe is the gold standard. We are not trying to replace those methods. What our goal is to see if we can find a way to be a comparable technology to save the analyst time and money when quick measurements within the process are required to take the next step. So I'm not talking about GMP release testing. I'm simply talking about a better analytic tool to make quicker and more accurate decisions. In order to do that, we needed to see, based on slope spectroscopy, if we can actually quantify a change of absorbance from a perceived full capsid versus a perceived empty capsid. And as the graphs and slope regressions confirm, we are able to do that. In the first test that we ran, we were able to compare slope spectroscopy's technique versus qPCR and ELISA and come within 9% difference of the 2 results combined. The main difference being is that all of the processing and testing time took the better part of a week, while the SoloVPE test took 5 minutes and arrived at the same result. The same technique can also be used in our inline solution, the FlowVPX. where we are monitoring viral titer over time. It's the same technology, just inline, so we're talking about adding kinetic software to collect data at the given time points the user inputs. In this graph, you can clearly see the stages of concentration up to diafiltration starting, and able to take these time points and quantify a viral titer concentration over time. And as you can see, compared to the expected offline values, we align very well. In this particular case, using the FlowVPX technology, there's zero sample manipulation since the entire process is done in line. We then took this a bit further and wanted to work with a company that's able to provide certified NIST traceable standards related to AAV material. The first demonstration of the technology was done at Vigene Biosciences, where we took some of their in-process samples for both AAV8 and AAV9 and compared the SoloVPE values versus their expected values. If you look at the percent difference column, we align very well to their given processes. What's more interesting in this data set is in the AAV8 empty slide, you can see we arrive at a 27.6% difference. However, they also arrived at a 15.4% difference. This was able to confirm that they truly did not have an empty capsid and potentially still had DNA left within the process. So not only did they accept this data, but they then used it to refine their process to understand potentially how they were still leaving product on the table. The following step was purchasing their NIST traceable standards, which are off-the-shelf items, to understand just how well the Slope Spectroscopy methods compare to a certified standard. We looked at 3 different serotypes, both full and empty, and when comparing viral titer concentrations, we were well within the plus or minus 40% for the ddPCR and ELISA method, with an average being about 7.5% for ddPCR and 3.7% for the ELISA portion of the assay. This confirmed 2 things. 1, we can still be equivalent in the process, and 2, our technique using slope spectroscopy is serotype independent. The last set of data that I'm showing you was from the Bioprocess Technology Institute in Singapore. We purchased certified material through them in 3 different batches at 5 different dilution steps. So we wanted the samples prepped at different concentrations and certified and tested using 3 different technologies: qPCR and ELISA, ddPCR and ELISA, and dPCR and ELISA. The 3 batches showed very consistent data from batch to batch and, more importantly, test to test. So a summary of this data represented here, whether we were compared against qPCR, ddPCR, or dPCR, we had equivalent results. We also tested this method versus TEM and got similar results as well in the same scope of percentages versus the expected values. In summary, we've now given you a tool for in-process screening. This method can be applied to all the areas where you see in green for a quick way to understand exactly what's in your process within 1 minute at any given time. To repeat, it is our goal not to replace the current techniques and processes that are being used in the industry. Our goal, in some cases, is to be comparable, and be comparable enough to allow you to make real-time decisions that are equivalent to any of the given methods that are currently being used. For viral titer concentration, screening from E to the 11th and higher has been a viable solution for all customers and certified standards we've measured over the last year. We've now been able to give a rapid and highly reproducible method for in-process screening related to AAV viral titer. It's quick. We're talking dual multi-wavelength analysis that typically takes less than a minute, and easily transferred due to the significance of a robust 21part11 software package. To summarize, we are committed to your success. you truly have a partner with Repligen within implementation of this technology in your environments. We have found that the closer we work with scientists in the lab in providing our level of expertise related to method development and ultimately method validation, provides a sharing of responsibilities between the two parties. It is in our best interest to set you up for success. We encourage everyone that's looking to implement Slope Spectroscopy as their platform method to work with our groups in order to give you the best chance of success and ultimately expedite the entire process of method development and validation. It is our goal that with both technologies for the at-line and in-line, we are giving you two analytic tools to properly monitor, quantify, and make real-time decisions on your processes where UV can be the preferred method for your particular product. The functional alignment between all groups within the process has been critical to our success. Working with analytical groups to help define the method, process development groups to understand the scope of the project, help feed that information that ultimately gets to forms of manufacturing and QC to make sure that all parties are not just using the technology, but can all benefit from adoption of a platform technology that carries all of the benefits related to accuracy and repeatability. I thank you for your time, and I think we're ready for some questions.
Brilliant, very nice presentation, Joe. So now we'll move on to the Q&A, as you said. So if any of the audience have a burning question about Joe's presentation or any of the topics raised, you can submit that via the Ask a Question box. Please do so. Okay, so first question that I've got here, Joe, is regarding AAV concentration, full empty ratio. I would imagine that the technology could measure partials, is that right?
Unfortunately, no. UV is not a selective enough technology to distinguish a partial versus a full, rather than just the total content of what is. So you would certainly see a change in concentration without doubt, but being able to properly define it as a partial, or if there's residual impurities within the sample, again, it's not really the job of UV to distinguish exactly what's what.
Okay, good to know. And are there any applications for CAR T processes of this technology?
Currently, not yet. I would say it's something we're not focused on short term. I can tell you that if your current method is a UV-based one, then yes, you can certainly apply slope spectroscopy to that, but our current focus is not at that step yet.
Thank you. And is the SoloVPE system and software fully GMP compliant? How are the system and software qualified, and what type of support is provided for qualification?
Sure. Every new system is purchased with an IQ/OQ, so it is fully installed and qualified, and then the software package that is provided with the system is GMP capable, so that's how we usually say that all the tools are there, and we either offer a software validation service, which will validate the software for you in GMP conditions, or work with your current IT team to see exactly how the configuration should be put in place for your specific group.
Brilliant, thank you. And can the SoloVPE be used to test incoming plasmid product as raw material to check purity levels?
Short answer is yes. Again, for ours, our approach is, again, it has to be purified material. So we are not looking at anything from the harvest. It has to be purified.
Absolutely. And how does the UV distinguish AAV from host cell DNA? from the harvest?
Right. So it's a multi-wavelength slope analysis. So we're looking at the relationship between the two wavelengths of interest and then doing a complex equation based off implementing slope-based extinction coefficients or published extinction coefficients from literature to be able to calculate the concentration.
Thank you. And can the SoloVPE method be used to for release of GMP finished drug product as well?
Yes. So that's where the majority of our business is in GMP environments related to drug substance or drug product.
Brilliant. And how long does it usually take for the SoloVPE system to be installed and validated for PD work?
So from our perspective as the vendor, for our side to complete IQOQ training and software validation, if needs to be, typically that's on average 1 to 2 weeks. The real work starts obviously with the detailed plans for how each group will implement it. So depending on how many methods are going to be validated could depend on the scope of work planned for each system. I would say on average, based on all of our experience, when the project has the appropriate time and resources to commit to, we typically see a complete method validation and implementation of our technology probably within 6 to 12 months. However, the majority of that is not relying on us as the vendor, more the resources of the companies that we're selling to.
Brilliant, thank you. Can you talk about the validation process and the validation support that Repligen provides?
Sure. So our applications group within bioanalytics applications has post-sales responsibilities with every system sold into the market. So you will be paired with one of our application specialists to understand exactly how you will be implementing the technology and then the associated method development and validation support in order to get that implemented within your organization. So you have essentially one-on-one custom support in order to help you through any SoloVPE project.
Thank you, Joe, for answering those questions. I think we'll wrap up there for today. Any questions we didn't get to, we will reply by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. I think all that's left is to thank Joe once again for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.